This video is going to discuss what's known as the IMViC panel. And it's called a panel because you have to do each of the four tests all together. You never just pick and choose one of them to do. And we use the IMViC panel to help differentiate our EEC group, the group of organisms that can ferment lactose. So we call this the IMViC panel because the four tests are Indole, Methyl Red, Voges Proskauer, and Citrate. So if you put the first letter of each of those tests together, you get IMViC. So all four of these tests are differential tests. So we are looking to see if an organism can do a certain process or not. Does it have a certain enzyme or not? So again, differential means anything can grow, it's just going to grow differently, or we'll be adding reagents or chemicals to the media after incubation in order to detect whether or not that process has occurred. So pay attention to this table. Make sure you know what the I, M, V, and C stand for, what type of media it is. Notice that 3 out of the 4 are broths, but for the citrate test, we use a slant. And make sure you understand that last column, because the column is called differential based on, but another way of saying that is the purpose. So the purpose of the Indole test is to determine whether or not tryptophan hydrolysis occurred. And if so, that means both pyruvate and indole were produced. Please note that pyruvic acid is the same thing as pyruvate, which we talked about back in fermentation. I'm sorry, in respiration. Okay, so let's move on to our next slide and start diving into the details of each of these tests. So first up is the Indole test. And this is a broth media that contains tryptone. Because remember, we're trying to determine if the amino acid tryptophan can be hydrolyzed. So that means we have to have that amino acid tryptophan present in the media. And so we refer to that particular type of broth as a tryptone broth. And in order to tell if tryptophan was hydrolyzed into indole and pyruvate, we have to first get bacteria on our inoculating loop. We swish it into the broth and we put it in the incubator and let it incubate overnight. We come back the next day and we add Kovacs reagent. And Kovacs reagent specifically reacts with indole. So if indole is present, the Kovacs reagent will react with that indole and you'll see a pretty red ring at the top of the tube. Now, sometimes, like you see in the picture, the rest of the media may become red, but that's not what we're looking for. We're specifically looking for the presence of the red ring at the top. So if the media changes colors and we see that red ring at the top, it's a positive reaction. If there is no change upon adding the Kovacs reagent, then it's a negative result. Please remember that anytime there's a color change, that is indicative of a positive reaction. If there's no color change due either to inoculation or adding some sort of reagent, then that means nothing has happened. Nothing has changed since putting in the bacteria, so that's a negative reaction. On the left side, where you see the red ring, which is a positive reaction, we have to think back to what is this telling us? What are we trying to figure out? And we know that indole is formed as a result of breaking down tryptophan. And on the right, it is not. Moving on to the M in IMViC, it's going to be the Methyl Red test, which is looking to see whether or not an organism can perform mixed acid fermentation. And it's called mixed acid fermentation because it produces a lot of, or a mix of, acid byproducts as a result of fermentation. Remember with fermentation that the pH of the environment will always go down. So we're going to have a pH indicator called methyl red that is already included in the media from the beginning. We don't, that's not true, sorry. Let me start that one over again. So you get your methyl red Voges Proskauer broth. Notice that both of the M and the V tests, methyl red and Voges Proskauer, they use the same broth. Okay, so we call it the MRVP broth for methyl red Voges Proskauer broth. You get the bacteria on your loop, you swish it into the media, you incubate it. When you take it out of the incubator, you add methyl red. So we call it a reagent, but it's also a pH indicator. Okay, so we add the methyl red after we take the organisms out of the incubator. And if the media in the test tube is acidic, then the methyl red will cause all of the media to turn red, like you see on the left-hand picture. And if fermentation did not occur, meaning the pH is not acidic, then adding the methyl red reagent won't do anything. So there will not be a color change, and the media will remain that kind of copper amber color that you see on the right. All right, so again, notice that the medium is called the MRVP broth. Okay, so the MR was for the methyl red test that we just talked about. Now we're going to talk about the VP portion, which is the Voges Proskauer. So we call this media a combination media because we can do a combination of tests with it. So it's the same media used for the methyl red test. Okay, so you grab the same broth. But of course, it shouldn't have anything growing in it. It's just the same nutrients and ingredients and things like that. And what the VP test is looking to find out is whether or not an organism is able to produce acetone. As I've already mentioned, when fermentation happens, the pH can become pretty acidic. And so oftentimes, organisms will produce acetone as a way to prevent the media from becoming too acidic. It's kind of helping to neutralize that media. And so acetone production is just kind of like this way to balance things out. And it also turns out that acetone can be used as a form of energy storage for some bacteria, and other organisms actually convert acetone into acetyl-CoA, which may sound familiar because that is the molecule that goes into the Krebs cycle during respiration. So to do this test, you take your MRVP broth, you get bacteria on your loop, you swish it into the broth and you incubate it. When you pull those tubes out of the incubator, they're going to be kind of that coppery color that you see on the right. You're going to add reagents Barrett's A and Barrett's B. And then you're going to just gently shake the test tubes. And you're going to have to do this periodically for up to about 30 minutes because just sometimes that color change isn't immediate. So you'll add the Barrett's A and B, you'll gently shake it, and then you'll normally just set the tube off to the side and keep working on other things. And just periodically, every few minutes, gently shake that tube again. If you eventually get a red color dispersed throughout the media, then that's a positive result. But notice in the picture, you see two different positive tubes. Sometimes the red won't completely disperse throughout the tube, and that's perfectly fine. Other times it will. It just kind of depends, okay? But it is going through at least about half of the media. And over time, if you let it sit, the media will darken. So it's important to be patient in giving it up to 30 minutes to change colors, but you don't want to wait hours because eventually the reagent will cause the media to darken no matter what. So you don't want to get a false positive there. And then as you can tell from the center picture, right, it's making me hungry just looking at it. Acetone can be derived from yeast and used as an artificial butter flavoring, which I'm grateful for because I love butter. All right, so let's go ahead and wrap up this video with a talk on the citrate test. And for this test, we're trying to figure out if an organism can use citrate as a sole carbon source. And in order to do that, it has to have two different enzymes, citrate permease and citrate lyase. So we're actually looking to see, does an organism have these two enzymes? If they do, then they are able to use citrate as a sole carbon source. So always remember to think about the enzymes and incorporate those. Those are always important. So this media, normally when we just make it and you haven't put any bacteria on it, it's this pretty kind of dark green slant. So you always inoculate a slant the same way. So you put your bacteria in or on the media, and you are going to incubate it. When you take the media out of the incubator, you look to see what color it is. If it's still green, then the organism was unable to use citrate as a sole carbon source. It does not have citrate permease or citrate lyase. So again, not using citrate as a sole carbon source. However, if you take the media out of the incubator and it's this pretty, it's kind of a deep royal blue color, then that means the organism does use citrate as a sole carbon source. It has citrate permease, which takes citrate into the cell, and it also has citrate lyase, which then allows the organism to break down that citrate and extrapolate those carbons. Because we're looking at an immediate color change, that means we have to have some sort of indicator in the media, and this indicator is bromothymol blue, and at basic pHs, it is going to change from the starting green color to a blue color. So the process of using citrate as a sole carbon source is going to cause the environment to become alkaline. All right, so let's take a look real quick at some of the results you might get if you do this in the lab. So E. coli will give you these results for the IMViC panel. So think about what this tells you about E. coli as an organism. And then you'll notice two of our potential unknown organisms give the same IMViC panel results. So take a look at them, consider what observations you're making, what are the results, and what does that mean in terms of an interpretation. All right, y'all, let me know if you have any questions.